Good morning, madam. Can you hear me?
Good morning.
Okay. You hear me okay, right?
ఓకే అండి. జస్ట్ నేను కొద్దిగా వెల్కమ్ అడ్రస్ చెప్పేస్తాను.
Sure, sure.
ఇన్వైట్ చేస్తాను.
No problem. మీకు వినపడుతుంది కదా?
వినిపిస్తుందండి, మీ వాయిస్ క్లియర్ ఉంది.
క్లియర్గానే ఉంది కదా? ఓకే, గుడ్ గుడ్. ఒకసారి చెక్ చేద్దామని.
Respected Honorable Vice Chancellor, Sir, Dr. K. Dhananjay Rao Garu, esteemed guest speaker of today's Global Connect program, Dr. Addepalli Balasubramaniam Garu, respected university officers, Dr. P. Prasanna Kumar Garu, Dean of Horticulture and Dean PG Studies, Dr. S. Surya Kumari Garu, Director of Research and Controller, Dr. M. Madhavi Garu, Registrar, Dr. Ruth Garu, Director of Extension, Dr. S. S. Vijay Padma Garu, Director P and MESL, and Controller of Examinations, Dr. Madhumati Garu, University Librarian and Dean of Student Affairs, all the Associate Deans of Constituent Colleges, ADRs of Coastal Zone 1 and 2 and Rayalaseema Zone, principals of Horticultural Polytechnic Colleges, heads of the research stations and KVKs, all the teachers, scientists, and rapporteurs of today's program, Dr. C.H. Nalini and Dr. K. Archana, and my dear students, a very and hearty warm welcome to all of you to the 30th Global Connect program of Dr. YSR Horticulture University. Your participation makes this forum vibrant and meaningful. Global Connect program has become a flagship platform of our university to bring together all the teaching faculty and students to deliberate on emerging trends in horticulture. Reaching the 30th edition is itself a milestone and reflects the commitment of Dr. YSR HU to knowledge sharing and global collaboration. This program is being organized under the able guidance and the leadership of our Honorable Vice Chancellor. We are privileged to have our Vice Chancellor, Sir, as the President of this program. Sir, your vision and constant support have been the driving force behind taking Dr. YSR HU to greater heights. However, today, Sir is, I wish to inform that our Honorable VC could not be present physically with us due to unavoidable official commitments. Sir has joined online to preside over this program. Then we are indeed honored to have Dr. Addepalli Balasubramaniam Garu, Director and Consultant Scientist, University of Cincinnati, Ohio, USA, as our guest speaker. So now I will go through the brief biodata of Dr. Addepalli Balasubramaniam Garu. So Balasubramaniam Garu did his BSc at Agriculture College, Bapatla, followed by MSc and PhD in biochemistry at IIRA. He initiated work on antiviral proteins that impart immunity to plant viruses for biotechnological applications. Later, he worked on induction of disease resistance responses by yeast poly-A binding protein expression in plants. Other research includes gene silencing, mRNA polyadenylation mechanisms at University of Kentucky, Lexington, USA. Afterwards, he demonstrated the utility of the enzymes purified from cucumber and bittergourd seed for characterization of RNA therapeutics at University of Cincinnati, Ohio, USA. Currently, he is working at Waters Corporation, where he is involved in analytical tool development, target reagent enzymes for critical quality attribute analysis of cell and gene therapy products. Sir has undertaken many research projects like cloning and characterization of antiviral proteins from non-host plants at IIRA New Delhi. And at the University of Kentucky, he had undertaken 4 research projects: disease-resistant properties of tobacco cultivars that express the E. coli genes, controlled expression of L-stin-like genes conferring broad-spectrum pathogen resistant in plants, functions of multiple poly-A polymerases in Arabidopsis, and Arabidopsis polyadenylation factors, subunits, mutants, and protein interaction networks. And from University of Cincinnati, he has undertaken projects like mass spectrometry of ribosomal RNA protein interactions and mass spectrometry of modified RNAs, and also he was the recipient of awards like ASPE and plant protection gold medals in BSc-AG, gold medal in MSc-AG, and best poster awardee in Rust Belt RNA conference. He has guided more than 60 undergraduate research works, 1 master's student, and 7 PhD research projects. He had about 63 research articles published in national and international journals. And he is a member of American Chemical Society, American Society for Mass Spectrometry, and American Association of Pharmaceutical Scientists.
DIP program.
So sir will be delivering a lecture on today the biochemical and biotechnological approaches to mitigate post-harvest food losses and improve the shelf life. Sir, your vast expertise and global insights will greatly inspire our faculty and students. Now I request Honorable Vice Chancellor Sir, Dr. Dhananjay Rao Garu, to deliver his presidential address.
Hello.
Sir,
Good morning. Good morning.
sir, I request you to please deliver the presidential address, sir.
Yeah. So, welcome to Dr. YSR Horticulture University. Sir, good morning, sir. Subramaniam, Dr. Subramaniam garu, good morning.
Good morning, sir. Thank you. Thank you
Welcome
for the
to
invitation.
welcome to Dr. YSR Horticulture University 30th Global Connect Series, sir. Thank you for
Thank you.
accepting our invitation and participating as a guest speaker today in Global Connect program. And we have started 2.5 years back this Global Connect series at Dr. YSR Horticulture University when I was Director of Industrial and International Program. And I am very happy that participate in 30th Global Connect series conducted by Director of Industrial and International Program, Dr. YSR Horticulture University, Dr. T. Padmalata Garu. And today guest speaker, Dr. Addepalli Balasubramaniam Garu, Director and Consultant Scientist, University of Cincinnati, Ohio, USA. Welcome, sir. And this program started to educate our faculty and students what is happening in the world and the international level to update the knowledge in horticulture and other allied subjects of horticulture. And the topic is biochemical and biotechnological approaches to mitigate the post-harvest food losses and improve shelf life. You all know that this is the major problem in horticulture crops, that is post-harvest losses. The fruits and the vegetables are very perishable in nature, and a lot of 20 to 30% are going waste. And we are very happy that we are knowing biochemical and biotechnological approaches to mitigate the post-harvest food losses. You all know that so many after harvest, so many biochemical changes are occurring, and due to that ripening and senescence of fruits and water losses from the fruits and mechanical injuries and microbial decay. So many things are happening. Due to that lot of wastage after harvesting during storage, during transportation, and many losses, 20 to 30% losses in fruits and vegetables. And whatever we have developed so much technology, but this is very important that the biochemical and biotechnological approaches. And distinguished guest speaker and all my esteemed university officers, faculty members, scientists, researchers, students, and other participants, a very good morning to everyone. And it gives me immense pleasure to welcome you all to the 30th Global Connect series of Dr. Y.S.R. Horticulture University. This lecture series reflects our commitment to connect with globally renowned scientists promoting excellence in horticulture education and research. Today topic biochemical and biotechnological approaches to mitigate post-harvest food losses and improve shelf life. A highly relevant in the present agriculture scenario. Despite remarkable progress in crop production, post-harvest losses continue to remain one of the biggest challenges. For a horticulture rich state like Andhra Pradesh, minimizing these losses is an important as increasing production. Today, modern science offers transformative solutions. Technologies such as AI and machine learning, computer vision, hyperspectral imaging, digital twins, blockchain-based traceability, RNA interface, and CRISPR gene editing, edible bio coatings, smart packaging, cold plasma, pulsed electric field, high pressure processing, ozone treatment, and waste valorization are revolutionizing post-harvest management. Many technologies have come today, that latest technologies, and we are going to listen from our expert. By integrating this next generation interventions with modern digital innovations, Dr. Y.S.R. Horticulture University can pioneer a smart technology-driven post-harvest ecosystem that enhances shelf life and farmers' income. Today, we are privileged to have Dr. Addepalli Subramanian Garu as our distinguished speaker, and moreover, sir's native place is Ongole, and moreover, he is our man. And sir completed BSc Agriculture from Bapatla and won the prestigious ASPE and Plant Protection Gold Medals before earning MSc and PhD in Biochemistry from Indian Agriculture Research Institute, New Delhi. His research career spans several internationally reputed institutions, including the University of Kentucky and University of Cincinnati, USA. He has managed multiple high-value research grants from the USDA, NIH, and DBT, published 63 high-impact papers, guided more than 60 research scholars, over 1,500 citations, and H-index of 24. H index of 24 is the highest rated, and we feel proud of you, sir. And his profound experience, expertise spans plant virus resistance, gene silencing, mRNA, polydenylation mechanisms, RNA biology, and analytical biotechnology make him as an ideal global expert to guide our perspective today. His expertise perfectly aligns with today's theme, and I am confident that his insights will ensure our scientists, faculty members, scholars, and students. I encourage all the participants to actively interact with our speaker and make the best use of this opportunity to learn from this vast international experience. I express my deepest gratitude to Dr. Balasubramaniam for sharing his invaluable time and global expertise with his peer back home. I also appreciate the efforts of Director of Industrial and International Program for organizing the important Global Connect series. With these words, I wish the program every success and enlightening and fruitful learning experience for all. Thank you. Thank you all. And you interact with our expert and have many any doubts, queries, and discuss and know many things from our expert. And thank you, Jai Hind. Thank you, madam. And you
Thank
can
you very
proceed.
much, sir.
Thank
Thank
you. You
you
can
very much,
proceed.
VC, sir.
Yes,
Even though
yes.
you are not present physically, through online, you have given
Yeah.
your presidential address. Sir, now let this session be fruitful, interactive, and lead to new ideas for research and extension in horticulture. Now,
Yeah.
I request Dr. Addepalli Balasubramaniam Garu to present the topic that is biochemical and biotechnological approaches to mitigate post-harvest food losses and improve shelf life. Sir, Balasubramaniam, sir, please
Yeah.
share your.
Yeah. Sure. Can you see my slides?
Yes, sir.
Okay, thank you. First of all, thank you so much for the invitation, respected Vice Chancellor and university officials, students, ladies and gentlemen. It's a privilege to talk to your own people. So I belong to you basically. So I come from Bapatla Agricultural College, and although my native place is Ongole, I valued my education in Bapatla and IARI very much, and that has helped me a lot to progress and make inroads into biotechnology, biochemistry research in United States. So it's a privilege and honor to talk to you guys and present things that are related to the post-harvest technology. So when I joined IRI, my first preference was plant breeding, but then I went to biochemistry. And then, after joining IRI, I realized that post-harvest technology is another important area where you may have a bumper harvest, but if you don't manage the crops and vegetables, people incur losses. So it's a lost income. It's a double whammy on both sides. So I realized that I had a friend who did PhD in post-harvest technology, and I interacted with him a lot while I was in the IRI. So I thought this is a good topic to choose and discuss various biochemical and biotechnological approaches that would prevent or at least minimize the post-harvest losses. So Honorable Vice Chancellor has touched upon a lot of topics. Actually, he gave a overview of what I'm going to talk about. So, he's highly knowledgeable, and you guys know a lot of things. I'll just try to give a overview, and then I'll encourage any, discussion. If you have questions, we could talk about it. and so on and so forth. So let's move forward. So this is my overview of presentation. So we'll look at the post-harvest losses that are happening in India and worldwide, how much loss is happening, and the reasons for post-harvest losses, right? So a couple of things we'll discuss about that, and then the traditional approaches, right? And they're effective, but sometimes they not be sufficient. So that's why you need biotechnological means, and biotechnology comes from biochemical knowledge. So the biochemistry, is the first thing. Once you know what are the pathways that are involved in, fruit ripening and losses and so on and so forth, then you could devise biotechnology means. And today I'm going to talk about two technologies: RNA interference and genome editing. So, just giving an introduction. So, the fruits and vegetables are considered as perishables, but why? Because they have short shelf life. They are living, so they undergo lots of physiological processes, including respiration and water loss, and so on and so forth. So, physiological reasons or primary reasons why these fruits and vegetables are considered as perishables, and they have short half-life or shelf life. Now, you may experience the loss in terms of the quality or in terms of quantity. So, customers prefer the fruits and vegetables that are firm in texture, but once there is a, enzymatic process that softens the tissue, then the fruits and vegetable will lose the value, right? And then they are also susceptible to biotic factors, pests, fungus diseases, bacterial infections, viral infections, so on and so forth. And then you may also subject to damage during transportation and handling, right? So you may have qualitative loss and quantitative loss. I don't have to emphasize all those things here, but when you look at the extent of losses, they may vary, 7 to 25%. Again, depends on the location, depends on the time of measurements and so on and so forth. So you have losses, in percentage wise, 7 to 25%, both in India as well as in, worldwide, right? So there are some references where it says 30 to 14%, but again, it's the time of the year and how these metrics are calculated. It may vary from place to place. Sometimes it may be even 50%. So depending on a location and the time of harvest and so on and so forth. So, and then in terms of the actual amounts, the absolute amounts, you may see 20,000 tons to 80,000 tons. Right, so that much food loss is happening. It's basically a loss for everyone, primarily for the farmer. The income is lost because of these losses. Now, what are the reasons for these post-harvest losses? So, as I said, the primary reason is physiological and biological reasons. These are living things. The fruits and vegetables are living things. So that means they respire, they exchange gases, and there would be water loss when the temperature is not right. And then the hormone is continuously happening. So hormone synthesis is happening, ethylene, which is a ripening hormone, can induce the generation of certain enzymes that would degrade the cell walls, tissues, leading to softening. And then in that case, the other enzymes, lipids, the enzymes will oxidize the lipid, so that would cause putrefaction and browning, right? Browning, hydrolysis, and so on and so forth. So you have these things happening, and that makes it easy for the microbial organisms to infect and cause further decay, right? And then, so these are the biotic factors, and you have abiotic factors like temperature, humidity, control or lack of it, and then mechanical damage during transportation. So all these factors, multiply the losses during the post-harvest handling process, right? But then how do we, prevent these losses or at least minimize them, right? So there are two different approaches, physical methods and chemical methods. So as you can see in the chemical approaches, you could use certain acids, essential oils, or chelating substances, or edible coatings. So basically, you are making a barrier so that the fruit's physiological process is controlled, or at least you have a handle on it, so that you prevent the water loss, or reduce the ethylene production, or inactivate the enzymes that are involved in browning process or softening process, right? And then you have physical methods, the heat treatment or irradiation. So we have seen here, in Western countries, the heat treatment is commonly followed to increase the shelf life, and we'll see how the advantages of the heat treatment. And then you also have irradiation through gamma rays, electron beams, X-rays, and so on and so forth. But this one is the radiation is not extensively used compared to the heat treatment. Now, let's look at the ripening process and the ripening hormone. So you have ethylene produced from methionine. Methionine is an amino acid, and it's an essential amino acid. It has a sulfur in that. It's when you say essential, it's required for the body growth. And plants, of course, they synthesize their own amino acids, and then it's converted into SAM, acetylmethionine, and then amino cyclopropane carboxylate, and from which ethylene is synthesized. So you have this ethylene synthesis pathway, and then that ethylene leads to the ripening process. Now you have a number of what they call transcriptional factors that aid in the induction of these genes, meaning these more enzymes are produced when these factors are present in the cell, right? And that leads to the increased production of ethylene. But then you have other factors that would act as an inhibitor for the ethylene production. So they're indicated by the inverted T's. So calcium, low oxygen, and then you have aminoethoxy, vinyl glycine, aminoxyacetic acid, so on and so forth. So these compounds, they inhibit the enzymes that are involved in ethylene production, right? So thereby, you could slow down the ripening process, right? And during this ripening process, what happens is sugars increases, and the color changes. So some enzymes are activated compared to the unripe situation, right? And when more sugars are produced, then microbes, what we call bugs, so they microbes, basically, they infect the vegetables or fruits, especially when the sugar levels are higher. So that leads to increased rotting of the vegetables and fruits. So you have both stimulatory processes that make the ethylene hormone, and you also have opportunities to downregulate or lower the production of ethylene. Now, one way to do that is, temperature exposure or heat exposure I was mentioning about, right? So when you expose the fruits and vegetable to, say, vapor dip or hot air treatment, right? So when that happens, you basically lower the enzymatic reactions, right? And you also reduce the microbial load and insect infestation, and you inactivate the hydrolytic enzymes. So these hydrolytic enzymes are responsible for cell wall degradation, right, and softening of the fruits. And you also reduce the post-harvest diseases like stem rot, arachnose, and so on and so forth. So, and of course, you maintain the appearance, the texture of the fruits, you maintain, so the customers will can easily buy those things. So basically you increase the shelf life when you expose to the heat, hot air treatment, or in warm water dip. So in the right side you have some examples where this heat treatment was implemented or is being implemented for mango, papaya, citrus, strawberry, and even tomatoes, right? So this is a very well followed practice to reduce the post-harvest losses, right? And sometimes you may have a packaging of the fruit, right? So where you could modify the atmosphere and humidity. Again, for the storage, you need to maintain two conditions, temperature and the humidity, right? They are the critical factors for optimal storage, right? So by packaging, you could control those things, especially the humidity, and that will regulate the gases exchange. So that would basically carbon dioxide increases and oxygen exposure is not happening that much. So that's why you could keep the fruit or vegetable in desirable condition, so that it doesn't ripen too much, or it doesn't become soft. So now here are some examples where different crops, fruits, vegetables are stored, the recommended temperatures and the humidity. Of course, I don't expect you to read through all these things. I'm just giving some examples. Suppose if you have a dried fruit, you could even store for 1 year, but if you have heat treatment or appropriate humidity, then you could store these things all the way up to 36 weeks. For example, apples, 34 weeks can be stored. So, and squashes, so they have 36 weeks storage. So you have a vegetable or fruit dependent temperature and humidity storage, so that you have a desirable shelf life before it goes to the market, right? Now, in spite of that, there is an inherent physiological processes going on that would ripen the fruits, and make them susceptible to the bacterial diseases. Now, conventional breeding is the regular approach that is implemented, but there are some challenges, especially while dealing with fruit trees, right? So you have long developmental cycles. So this breeding happens with the the flowering and fruiting. So sometimes it takes a lot of time. So selection process is too much. And then you have genetic and biological bottlenecks because of the selective breeding. So selective breeding led to a lowering of genetic diversity, so narrow genetic pool. So you may not have the desirable gene for breeding purpose, and you also have sexual incompatibility, right? So especially in Brassica family, right? So you have that self incompatibility. So that means the crossing is not feasible, right? And then some linkage drag. So some when you transfer a desirable trait, sometimes you may have undesirable trait coming along. So that's a linkage drag. And you also have environmental and phenotypic vulnerability. So you may see very good yield and desirable post-harvest behavior, but again, that depends on the phenotypic traits, and that phenotypic traits are subjected to the temperature, soil, or so you may not have a clear identification of the cause and effect situation. So that's why subjective selective selection and high failure rates. You have to screen a lot of plants to identify the right trait. So, and then of course you have customer, complaints. Sometimes you may have nice post-harvest behavior, but the taste may not be good, durability may not be, in the sufficient situation. So, and then yield drag. So you have all these challenges associated with conventional breeding. So that's where the biotechnological approaches come into the picture, right? So this is a overview of what biotechnology is employed to transfer certain traits, starting with molecular marker-based breeding approaches that are generally followed even before. So that those days enzymes were used, isoenzymes as a markers, but now genetic markers are used. And you have antisense RNA technology. We'll talk about that in detail today. And then you have mutation breeding, where the seeds or the plant material is exposed to the mutagens, and then you screen for the trait you are interested in, and then transgenics, right? But there is a lot of resistance to the transgenics because you may transfer some bacterial genes, unwanted genes. So that's why there is resistance to that. But there are approaches where you could, take away those unwanted genes through selection process, and then genome editing. So antisense technology and genome editing are the two basic things I'm going to talk about today. So you have these genetic engineering techniques, so CRISPR-Cas9, I will see what is that, but that will change or edit the genome, and you have zinc finger nucleases, transcription activated linked enzymes, and marker assisted breeding. So you have several examples where these techniques are employed to get the desirable post-harvest behavior, where one or two enzymes are, targeted so that they behave in an expected fashion. So now let's go into the details. The CRISPR and RNAi, so RNA interference are clustered, regularly interspaced sequences and palindromic repeats. So they have been employed in various fruit crops and vegetables where you could delay the ripening, you could develop oxidative stress resistance. You could control the lipid oxidation so that the rancidity won't be developed, right? And you can also use these techniques to fight a pathogen infection, right? Viral infections, prevent mycotoxin reduction, and again, viral infection in the livestock and aquaculture. So you have these approaches available to minimize the post-harvest losses. And let's go into the details, right? The antisense technology is also referred as RNAi or RNA interference. So we'll see what is RNA interference, right? So, but before going into that, let's recall our, genetics, knowledge of making proteins in a cell, right? So you have a double-stranded DNA. You have, when I say double-stranded, so you have two strands. Some direction is there, 5 prime to 3 prime, 3 prime to 5 prime, because they're complementary. Again, you don't have to remember that for now, but just remember that there are two strands of nucleotides in DNA. It's a storehouse of genetic information, and it is transcribed into RNA. So you make a single strand of RNA, right? So it's basically a representative of the DNA, right? Now, this RNA can be called as coding or non-coding. So when they say coding, so that means it can be translated into a polypeptide through ribosomes, right? And then if it is a non-coding, then it can become part of the ribosome. Ribosomes has RNA and protein, right? And then you also have other non-coding RNAs like transfer RNA, right? And then small nuclear RNA and so on and so forth. But the point is you have, a stepwise fashion where proteins are synthesized. And at each step, you could control whether the protein is made or not made, right? So that's why I have this trophic signal. So you could control these processes at the DNA level, right, to control the transcription, or even at RNA level. So RNA interference happen at the mRNA level, whereas the CRISPR-Cas9 system, that happens at the DNA level, but also uses RNA. We'll see more in the later slides. So how does the RNA works, right? So we talked about DNA, which is double-stranded, right? But in the RNA interference, you have two strands of RNA. So that's why it is called double-stranded RNA. And that double-stranded RNA is generated in multiple ways. You could make a transgenic and make the RNA, so which is a double-stranded. This is called a hairpin loop kind of RNA. And there is enzymatic machinery that would basically cleave the RNA into what they call small interfering RNA or siRNA, right? And then this siRNA has a sequence that is complementary to the messenger RNA. So you see this poly A tail, so that's messenger RNA. And when it finds complementarity, then a complex is formed. It's called RNA-induced silencing complex, that is called RISC, and then it will degrade the RNA, right? So this way it will degrade the RNA, and this also happens against virus diseases. So virus, when it infects, it makes double-stranded RNA, and then double-stranded RNA is recognized by these miRNA genes. That's called microRNA, and this microRNA recognizes the viral RNA, and then it'll basically cleave off the viral genomes, right? So you have various ways to lower the mRNA production in the cell so that the proteins are not formed, right? So there is a short interfering RNA. You could use one RNA against the target gene, or you could make pools of siRNA to target the gene, Or you could make a transgenic that makes short hairpin RNA and then siRNA and so on and so forth, right? So there are various ways. Let's not go into too many details, but if you have questions, I'll be happy to answer. So now, this is all good. This is happening in the cell, but is it practicable, right? Can it be practiced at the field level, right? That's the question. So the answer is yes, you could practice it at the field level, and I have some examples here. But of course, how you make the siRNA will, determine the cost of this usage. If you synthesize chemically, it is quite expensive to make a gram of that. But if you use cell-free enzymes, so there are some, Green Life Industry, so the biotechnology groups, they make these, siRNA in a pretty inexpensive fashion, just less than $1 per gram. Imagine that. So that means less than 100 rupees, you could make a gram of siRNA, right? And you could also make an E. coli and other organisms. Now, once you make this siRNA in a test tube, then you could spray it on the plant, right? You could spray it on the plant, and that siRNA is taken up by the plant cells, and it will control the either the bacterial gene expression or its own host gene expression, depending on the siRNA you are using, right? So you could make a transgenic and make the host to make the siRNA. That's one way. That's called host-induced gene silencing. Or you could make the siRNA outside and spray it. So that's called spray-induced gene silencing, right? So this way you could silence the genes that are involved in a ripening process or in the softening of the fruit tissue, right? So you have several examples here for SIGS, spray-induced gene silencing, right? That are implemented in variety of crops, right? And they have seen pretty, visible effects and important observations, gaining some advantage after harvest, right? So that is clearly implementable at the field level. That's the point I'm trying to make here. Now, so let's look at the, fruit rot, how this happens, how it is connected to the ethylene, right? So ethylene is called as fruit ripening hormone, but then once ethylene is formed, it can cause the induction of certain genes. When you say induction, so it make the genes to express, so make mRNA and proteins, and those proteins are enzymes. So those enzymes, polygalacturonase is one that is involved in softening of the fruit tissue. So you have all these enzymes that are listed here, and you could find it in your textbooks too. But basically, when those enzymes are generated, there is softening, and they are susceptible to the, handling, increased bruising, right? And then the pathogens can infect pretty easily, and enhanced water loss and so on and so forth. So you have all these disadvantages when these hydrolytic enzymes, what is meant by hydrolytic? Basically, they add water, right? So when they add water to the polymer, that is a condensed state, then you break down. So hydrolytic means breaking down of the cell wall, right? And the first example for RNA interference happened with tomato. They developed a variety called flavor saver. So where they basically introduced a gene, polygalacturonase gene, in a reverse orientation. Remember, we talked about 5' to 3' and 3' to 5'. So if the gene is in 5' to 3', then they made it in the 3' to 5' orientation. So when the orientations are reversed, they could come together and form a double-strand RNA, and that double-strand RNA leads to the inhibition of the mRNA production, and the protein is not formed. So that's why then the rotting of softening of the tissue was prevented, right? So this is how this antisense RNA technology works, and we're looking at one example. Of course, like that, you could target any enzyme that's involved in ethylene production, right? And then reduce the ripening. Now, here is one example where they're targeting a non-coding RNA, right? Remember, we talked about coding and non-coding RNA. Coding RNA, it makes the protein. Non-coding RNA may not make the protein. It won't make a protein, basically. But still, some long non-coding RNA, here example 1459. So, this one, is a positive regulator of ripening process. But once you make a siRNA or a deletion, so, there are two ways to control this. You could target siRNA, or you could edit it in that fashion, then you could control the ripening process. So what I'm trying to say here is, apart from the enzymes, there are non-coding genes that are involved in a ripening process. So how do they work? They work through the chromosome reorganization. So that reorganization leads to the increased synthesis of enzymes, right? So that's about antisense RNA technology. Now let's look at the CRISPR-Cas9 system, where you could do the genome editing, right? So there are multiple components in the CRISPR-Cas9 system. One is called single guide RNA, that's called sgRNA. It has two components, tracker RNA and CRISPR RNA. And then it also has a sequence called PAM sequence, and there is a protein called Cas9. So this complex is required for editing process. Now, how does the editing work? There is a sequence in the sgRNA that is complementary to the DNA, right? And then there is a PAM, which is not complementary. So that PAM is basically NGG sequence. It's called protospacer adjacent motif. So when the sgRNA binds to the complementary sequence in the DNA, the Cas9 acts as a nuclease. It will cleave both the strands of DNA, right? 3 base pairs upstream of the PAM sequence, and then that will introduce some deletions. That's called insertions or deletions. They're called indel mutations. And sometimes they could also be used to delete a gene or insert a gene, or you could even correct a faulty gene, right? So there are all these things are possible with this system. And we have our own example where, in DRR and then IARI, they used this CRISPR-Cas9 system to develop and release some varieties of rice that are tolerant to salinity, heat tolerance, and high yielding, right? They are not considered as transgenic because you are not introducing a gene, and there are certain rules on how you could call it as a transgenic or non-transgenic, but still you could it's similar to the conventional breeding, and then you could incorporate those traits, right? Now, what are the advantages of this CRISPR-Cas9 system? You could modify a lot of things. Plant architecture, of course, you could target fruit ripening pathways. You could incorporate disease resistance, abiotic resistance, carbon nitrogen metabolism, and even herbicide resistance. So the opportunities are like endless, and the scope is pretty vast when you employ this CRISPR-Cas9 system. So you have several examples that are already implemented where certain traits are modified by using this CRISPR-Cas9 system, right? And here in this genome editing process, you have certain steps. First, you need a RNA, guide RNA, single guide RNA that is complementary to the sequence. And you also use agrobacterium mediated transformation because you are doing genome editing, so DNA editing. So you could do it in the cells or you could do it in a protoplast. So there are various ways to do that editing. And then if you are doing in the cell, there are back crossing is done to remove the Cas9 or sgRNA gene, but you're left with the edited genome, right? So and then the people have made a number of Cas9 variants, so that you could do single base edition or you could do prime edition, so next generation breeding. So again, and then you could also have a protein that doesn't need the PAM, NGG sequence, in order to cleave the double-strand RNA. So there are a lot of things are happening where the Cas9 variants are made to improve the editing process. And there are examples that are happening with the tomato where you could modify the plant phenotype characters. You could incorporate biotic stress resistance, and then fruit nutrition and quality can be improved, and then of course abiotic stress tolerance. So there are several examples where antisense RNA technology as well as CRISPR-Cas system has been used to modify the plant traits. So to summarize, if you have all these areas, concentric rings, where the central one is the development of better shelf life, right? And then after that, we haven't touched this one. Sometimes you may have bitterness in the fruits and vegetables. You could delete those genes by employing this antisense or even CRISPR-Cas technology. And you could make climate resilient varieties resistant to abiotic stress. And then you could also, nowadays people are trying to use integrated AI-based tools so that you could accelerate the biotechnology methods, right? Of course, this is all the things that you could apply, but after this you have a bumper harvest. So what do you do with that, right? So they may slow the ripening process, they may withstand the biotic stress, but still you have a lot of things to do. So in that case, you could do the value addition, right? It is not a biotechnology means, but the biotechnological traits or biotechnological approaches can incorporate some traits that would add value to the fruits and vegetables, right? So you could do the value added products, and that means the increase in revenue, right? So, just like agriculture, horticulture is also an integrated approach. So it's not just one or other. It has to be all of them to be included so that you have a a high revenue and minimize the losses and better utilization of the crop produce. So with this, I thank you all for your kind attention, and I'll be happy to answer any questions you may have.
Thank you very much, sir, for your excellent and insightful presentation on biochemical and biotechnological approaches to mitigate the post-harvest food losses and improve the shelf life. Sir, you have very well highlighted the traditional and biotechnological approaches to mitigate the food losses, the physiological and biochemical causes controlling the ethylene production, and how modern biotechnological tools, enzymatic treatments, genetic engineering techniques, genes like gene silencing, genome editing through CRISPR and Cas9, how they can play a major role in reducing the post-harvest losses in fruits and vegetables. Sir, this talk has given both scientific depth and practical directions to the students and our researchers, sir. Now, I request the participants to interact with the guest speaker, raise your queries, and clarify the doubts. Any questions from audience? Please interact if you have any doubts. Ms. Subramanian, sir.
Yeah, feel free to ask any question. If I don't know, I can come back and let you know the answer.
Yeah.
Yep.
Sir, good morning, sir.
Good morning.
Please put
Go
on
ahead.
video. Who is asking the question?
Good morning, sir. Am I audible, sir?
Yep, I can hear you. Go ahead.
Thank you, sir. I am Dr. Sirisha, sir, a pathologist.
Yep.
Actually, sir, you have mentioned like shelf life can be improved and quality of the fruit ripening can be decreased. To how many days that can be decreased, sir? Or increased? Shelf life can be increased for how many days, sir?
Yeah, so it depends on the fruit and vegetable that you are targeting, right? And the enzymes you are targeting. So you have upstream genes, downstream genes, right? So, and what is the level of inhibition, right? So if you have a strong siRNA, for example, or even if you edit a gene, so how much editing success rate is there, right? So that would determine the increase in shelf life, right? So it's difficult to say how long the shelf life would improve. It depends on what extent that editing has happened, or the siRNA is able to control the mRNA level, right? So it could be 1 week, it could be more than 1 week. We have seen depending on the situation, right? It's a target dependent, situation, and the efficacy of the tools that you employ to reduce the expression.
Thank you, sir. Sir,
Yeah.
I have
Oh,
one more question, sir.
sure.
Any commercially available spray, double-stranded RNA sprays, sir, for crop
Yes.
protection?
Yes, there are spray-induced gene silencing. So there is a pathogen called botrytis. They targeted siRNA against this botrytis pathogen, and they reported a number of effects where the pathogen infection was completely reduced, and fungal infection is a fungus, right? So what they reported was if you include siRNA as well as fungicide, the effectivity of the fungicide was much higher, it seems, right? So you have a synergistic impact going on with the fungicide as well as the siRNA that is used, right? Sometimes you could use pools of siRNA to target the fungal cell wall synthesis as synthesizing enzymes, right? So that way you could inhibit the fungal infection, right? So like that, I'm just giving one example. Like that, there are several examples that are already demonstrated where you could decrease the fungal or viral infection, at the same time increase the shelf life.
Please mention about like commercially available double-strand RNA fungicides, sir. Commercially
Um,
available products are there, if any, available if any.
yeah, so I have a reference. I could forward it to you. I don't remember the exact brand name that they have used. I do remember the this one botrytis fungus, but I can let you know later.
Okay,
Sure.
sir. Thank you, sir. Thank you, sir.
Mhm.
Thank you for the
No
presentation.
problem. Yep.
Any more questions from the audience?
Sir, sir, good morning, sir. Very good, nice presentation, sir.
Thank
Here,
you.
here in Andhra Pradesh, there is more post-harvest losses due to post-harvest diseases in several horticultural crops, sir. Mainly in onion crop. After harvesting of onion crop, we cannot control this rot diseases, and also in mango commercially fruit crops also there is a stem and rot disease in mango and other fruit crops, sir. We can recommend only before harvesting only we can recommend fungicides, sir. After harvesting in mango, we can recommend hot water treatment, sir. Sir, if there is any this RNA sprays are commercially available, this double-stranded RNA sprays for control of these diseases in onion crops, sir. Is there any literature or available this RNA spray, sir?
For onions.
Onion, sir. Onion, it is a commercial crop, sir, in Andhra Pradesh.
Sure, sure.
After harvesting, there is no fungicide. We can recommend, sir. We cannot recommend any fungicide after harvesting, sir.
Yep. Yeah,
sir.
that problem is prevalent not only in India, but we also see it here in
Sir.
US. For example, US imports a lot of onions from Mexico and other nearby state countries, and we see that quite often, especially during winter season, right? January through April, even May, we see onions where the inside
Sir.
layers are softened, and you see fungus almost growing on the outside, the black
Yes, sir.
soot, right? Yeah. So that's a problem not only in India, it's everywhere, right? So it's a universal problem. That's what I'm trying to say
Yes,
here.
sir.
So have not come across any measure, but I will look further and
Yes,
then I'll let you know if there is some
sir.
available. But for the mango stem rot, yes, the hot water drip is pretty
Sir.
good. And yeah, so, but for
Sir.
onion, I have not come across any literature, but I'll let you know if I do that. So I have two things. I'll get back to you.
Okay, sir. Thank you, sir.
You're
Thank you
welcome.
so much, sir. For this is the right time, sir. I appreciate Dip Section Madam and a very nice presentation for arranging this Global Connect program, sir. Thank you so much, sir.
I'm welcome. It's my honor and privilege to talk to you guys.
Thank you, Ruth Madam. Ruth Madam is the Director of Extension, Dr. YSRHU.
I see. Okay.
Thank you, sir.
Sure.
Any queries from the participants? Any other queries?
Good morning, sir. Dr. C. Madhumati, University Librarian. Sir, apart from Flavor Saver Tomato, are there any commercially like CRISPR-rated crops or antisense technology developed commercially used crops in fruit crops, sir? Any
Yeah,
such type
so
of?
I showed a table where people have employed things. Of course, lots of those examples are in the field trials. Sometimes some of them are tested in the glass houses. Sometimes in the small even in the laboratory, but a number of them were also tried in the field level, and especially there is a company called Green Life Biosciences or Biotechnology. I can give you more details. So they are making this double-strand RNA, at a very low price. And they demonstrated that yes, it works on multiple crops, including, of course, tomato is the one. It's a favorite plant because it is easy to transform and easy to make transgenic and test it. It's a D-neutral plant, so that's why tomato has been used extensively compared to other crops. But they do have conducted these trials at the field level and demonstrated that it works. If you want specific examples, again, I'll get back to you on those situations. Sure.
Thank you, sir. A very nice presentation and very informative for researchers. And can we take up such type of works in India? And what are the facilities or infrastructure or funding required so that we can scientists can plan like such type of works?
Sure. Sure. One thing is, I showed you in DRR and IARI, they have done the genome editing where they may use this CRISPR-Cas9 system to, I think, delete a couple of or 7 base pairs in certain regions of the genome, and that led to increased tolerance to salinity and thermotolerance, and also
Nice.
increase the yield. So Fusa so yeah, it is possible, feasible. It can be done in India. It's not exclusive to any country. It can be done in any laboratory. So for that, what you need is you need to have nucleotide sequences. So DNA sequence is required, and you also have certain marker assisted knowledge. So the breeding program where certain traits are transmitted with the marker, right? So when you have that kind of knowledge, then it becomes easy to design a single guide RNA, right? So single guide RNA, you could design easily against those sequences, and then Cas9 can be made again. A lot of vendors are coming up, by technology companies are coming up where they make this protein and give it to you, right? You could make the RNA, you could make the protein and give it to you. What you have to do is inside the laboratory, you manipulate the genome, right? Either in the protoplast. So, we have these tissue culture facilities, right? So where you could grow the plants in the media, right? So you could also isolate the protoplast, and you could, in fact, transform these protoplasts with these Cas9 sgRNA complexes to do the editing process, and then you allow it to multiply and do the tissue culturing, right? So yes, it is feasible and possible. Only thing is you may need some guidance on designing a single guide RNA, right? And then identifying the right vendor that makes these proteins and RNA in a highly purified fashion, right? When you make these long sequences of sgRNA, the impurity levels are higher. So that's why, you know, you want to have at least 90% purity of the sgRNA and also the protein. So you have some homogeneity of these components, and then you could you know, manipulate the genome inside the laboratory, right, in the protoplast or even you know, small tissue samples, right? So yes, it is feasible.
Okay, sir. Thank you. Thank you very much, sir.
Yep.
Thank you, sir. Good morning, sir. This is Dr. Surya Kumari, Director of Research, sir. Sir, I have thank you very much, sir, for the excellent information you have given us regarding the topic. Sir, I have a small question, or I want a solution. So you have given many promising post-harvest technologies and have also given excellent results under laboratory conditions. But these face challenge in commercial adoption, right? In your experience, can you suggest or tell us what are the key barriers in translating these biochemical and technological innovations into cost-effective technologies, especially in a developing country like India, where to be adopted by the farmers or the horticulture industry, sir? There is one question from me, sir. And in this increasing temperatures, and as in a situation where we are at present facing the El Nino with erratic climatic conditions, so how can our post-harvest quality and horticulture produce? What are the interventions to be considered you suggest for promising shelf life and at this situation, sir? So first one is, how can we convert into a commercial technology? Can you please suggest us, sir, from the laboratory?
Sure. So let me reshare this slide that will give you some context here, and it'll also provide some inputs to the other questions previously asked. So let me share. So I think. Okay, so if you see RNA interference, there are a lot of situations you see in the control outcome measured. So there are laboratory studies, glasshouse studies, and field studies, right? So for example, in the second one, Solanum tuberosum, so the brinjal, so they did the studies at laboratory, glasshouse, and field studies, and you see the amount required 500 nanogram per microliter, so 0.5 microgram, and so 24 milligrams per liter is used. So I'm just giving one example, right? So like that, there are multiple situations where the field tests were done. Again, this is a simple snapshot of certain examples I'm showing. It's not a comprehensive, you know, literature I'm showing here. So I'm just giving a glimpse here, right? Now, to answer other questions, so what kind of components need to be made? So, and the cost, right? So here there is you know, so this table is somewhat incomplete. So what you need is biochemical know-how, right? So where you could make these enzymes, and these enzymes that are made you know, will synthesize the RNA. So you make a proprietary platform. So there are vendors, the companies that are coming up that make these siRNA, and you see the cost when they make these things, it's less than $1. Of course, when they sell it to the farmers or customers, it may not be $1, right? It may be $2 or even $5, right? So because they want to make profit, right? So this is the cost of making them. But the thing is, and this technology may be patented, right? But the basic ingredients, the basic biochemistry, and the know-how of making these enzymes is already available. It's a open source, right? So we just need to identify the person or a technologist who could do these enzymatic reactions clearly, right? So that means the biochemistry, we have to encourage people who can do biochemistry, right? Or, enzymatic synthesis, right? So then you could make this siRNA, quite good amounts in a cost-effective fashion, right? So again, so you want to identify the problem first, which one you want to solve, whether you have any traditional methods available to tackle that issue. If not, then you think about genetic means, right? So this is how you could do that. So whether it is a biotic stress, so fungal, bacterial, or viral, or is it an inherent physiological behavior, right? So once you know what is the problem and what's the situation, then you could devise the means, right? So again, it may sound fantastic, but it's becoming into the practice. It is coming into the practice quite fast, right? So there are a lot of agricultural companies, biotechnology companies that are involved in making these RNAs, siRNAs, in quite a good amount in a cost-effective fashion. I mean, we could do that too in India.
Thank you, sir. Thank you, sir. Thanks for the information, sir.
Sir, are there any commercial formulations available in the market, sir?
That's a very good question. So I think similar question was asked previously. I think there are. I can give you some examples later when after going through the website, and I will send you some details later on. Off the top of my head, I don't remember those commercial examples, but I can send that information.
Oh, thank you.
Mhm.
Thank you, sir. Okay, if there are no more queries.
Madam, this is Paratpara Rao, madam.
Yes, sir, please.
Good morning, sir. Balasubramanian, sir.
Good
I
morning.
have two questions. I have
Sure.
two questions regarding CRISPR-Cas gene editing technology.
Okay.
One is, so how far can we use this technology to improve the traits controlled by quantitative genes, polygenes? That is one question, sir. Second one is regarding in India, brinjal, one of the major vegetable crops, and
Sure.
fruit and fruit borer is the major issue. But there is a lot of opposition towards BT brinjal. BT brinjal, there is a lot of opposition. So what is the status of use of CRISPR-Cas technology to address this issue of shoot and fruit borer in brinjal?
Yep. Very good question. So the first one was, sorry, what was the first one?
For improvement of quantitative traits.
Quantitative
Like,
traits.
like polygenic inheritance.
Yep, right. So what happens in polygenic traits is that these traits are generally, of course, it depends on the plant, it depends on the crop. They're mostly linked. So you have linkage maps, And in rice, people, I talked to one of my friends who is working in Dow Agrosciences in Hyderabad. So he was talking to me. So he was saying they targeted a non-coding or intergenic region, right? So it's not exactly a promoter region, it's an intergenic region. And they edited or kind of deleted 7 base pairs in the DNA through CRISPR-Cas9, and that led to this improved yield and salinity resistant tolerance and thermotolerance. So what I'm trying to say here is identifying the linkage maps and targeting those genetic regions. Of course, there is no guarantee that you target one region and then it would work. There is no such thing. There are more failures than successes, but you got to start somewhere and make a hypothesis that, okay, so this is the linkage map, and this is how these things are transmitting from one generation to another. So is that part of the tolerance contributing system, or is it uh, not beneficial, right? So depending on that situation, then you could target, right, with CRISPR-Cas9 system. You could do the indels, insertion or deletion, or even you could change the genome through knock-ins, right? So where you have this homology directed repair, right, to knock in, to add a gene, right? So that's how you could do it. Again, I'm giving in a very broad sense how you could do it and how it was accomplished in rice, right? So that is feasible. Now, for the second question, that's really a tough one. So far, the examples I have seen is with microbial diseases, right? Not against the pests, right? So, or the insect pests, right? Now, there are other ways to do that. I've seen one technology called phase display, where they engineered the beetle resistance, right, through screening of various protein peptides, basically. So now, whether that would work, probably or not, I don't know. But this is a tough one because the insects, so they can bore into the fruit tissue, right? Whether it is whether it is firm or soft, they can go into they can bore into the fruit, right? Even an unripe fruit, right? So now to to develop resistance against that, it has to be thought in a different fashion. So like um, other chemical means maybe, or increase the tissue thickness, the enzymes that make it more resistant to the boring habit, or or add some chemical increasing enzymes that would basically repel the pest, right? So that kind of things may be feasible, but again, it requires a little bit of, or actually extensive literature review, and what kind of pest and how it infects or it bores into the tissue, right? So that kind of basic science studies would be required in order to implement a workable strategy. Yeah, I don't have a clear answer for you. Sorry.
Okay, sir. Thank you, sir. Thank you very much. Okay, sir.
Yeah.
Sir, good morning, sir.
Yep.
I have again another question, sir. What
Sure.
about the biosafety and biosafety issues regarding SIGS, sir? Are there?
Yep.
Yes, sir.
So, sure. So the regulatory compliance is a must, of course, when you are doing HIGS, host-induced gene silencing, where you are making transgenics. But if you are doing spray-induced gene silencing, they're transitory, right? So the RNA eventually gets degraded in the cell, right? So they are you have zero hazard or very minimal hazard or not much issues with the biosafety with the spray induced situation. With the host induced situation, yes, when you are making transgenic, there is a biohazard or biosafety issues, and that's why they are doing back crosses, right, to clear away the genes that are not required, like Cas9 protein or any other promoters that are expressed along with the gene. So they do the back crosses to remove those things and to answer the biosafety issue. So that's what they are doing. I've seen several examples doing that, especially if you do the crossing. But if you have self-incompatibility issues, then that's in that case you have to follow a different strategy. Yeah, I don't have a direct answers to this. I can give you some possibilities, but I can share some of the companies' information that are making these siRNAs and even CRISPR-Cas systems for commercial use.
Thank you, sir. On behalf of Dr. YSRHU, I thank you once again for accepting our invitation and sharing the global insights on this topic, sir. Now I request Registrar Madam to propose a formal vote of thanks.
Thank you.
Thank you, Madam. Respected Dr. K. Dhananjay Rao Garu, Honorable Vice Chancellor, Eminent Guest Speaker, Dr. A. Balasubramanian Garu, esteemed university officers, all the heads of institutes of Dr. YSRHU, faculty and scientists and students. Very good morning. At the outset, I sincerely thank our Honorable Vice Chancellor for his visionary leadership and continuous encouragement in organizing this type of academic programs that strengthen research, innovation, and capacity building in horticulture science. Thank you, sir, for extending your introductory remarks, sparing your valuable time in spite of your busy schedule. Today's lecture has been highly informative and intellectually stimulating. We have gained valuable insights into how biochemical and biotechnological approaches play an important role to effectively reduce the post-harvest losses, enhance shelf life, maintain nutritional quality, and improve the marketability of horticultural produce for extended periods. The most important aspect that caught my brain was that post-induced gene silencing, spray-induced gene silencing approaches that has been proved practical utility in many crops. But however, as Sir told, the efficacy, cost, regulatory clarity, and socio-environmental impacts definitely require further evaluation for broader use of these evolved techniques. And I hope these technologies will prove immense significance in the present scenario if research proposed in this direction at our university also, where reducing the post-harvest losses and ensuring quality produce are essential for increasing farmers' income and promoting horticultural exports. On behalf of Dr. YSR Horticulture University, I express our heartfelt gratitude to our esteemed resource person, Dr. Addepalli Balasubramanian Garu, Director and Consultant Scientist, University of Cincinnati, Ohio, USA, for sharing profound knowledge, rich experience, and practical perspectives. Sir, your inspiring lecture has motivated our faculty members, scientists, and students to explore innovative research in post-harvest science and biotechnology. I also express my gratitude to the deans, directors, all the heads of institutes, faculty members, scientists, and students for their enthusiastic participation, meaningful interactions, which made today's program highly successful. My special appreciation goes to the organizing committee, Dr. T. Padmalata, DIP and convener of today's guest lecture program, technical and administrative staff for their meticulous planning and dedicated efforts in ensuring the smooth conduct of this guest lecture. As we conclude this program, let us translate the knowledge and ideas shared today into meaningful research, innovative technologies, and practical solutions that will benefit our farmers, entrepreneurs, and the horticulture sector as a whole. With these words, I once again express my sincere thanks to everyone present over here for making this program a grand success. Thank you all.
Thank you,
Thank
madam.
you, sir.
Thank you very much, madam. With this, I declare the 30th Global Connect program of Dr. YSRHU closed.
Thank you.
Thanks, Andy Balasubramanian garu.
Sure, no problem. Thank you. Thank you.
Thank you, sir.
I'll get
Thank
back
you,
to
madam.
you. Yep. Bye.